Hi everyone. So we are on to the second unit for general biology, and we will now be focusing on the macrobiology part of plants. Now, at the end of this lesson, hopefully you guys will be able to identify the different plant cell types, describe the different plant tissues, differentiate meristematic and non-meristematic tissues, and explain how different plant tissues aid in growth and development of plants. Okay, so our first topic would be about plant cell types. Now, plants consist of several cell types, and this section will be tackling about the most common ones, which are first we have ground tissue cells. Now, ground tissue cells make up the ground tissue. Now, the ground tissue makes up the majority of the body of a herbaceous plant. It also consists of three main cell types, which are the parenchyma, the collenchyma, and we also have the sclerenchyma. So first of all, we have the parenchyma. Now, parenchyma cells are the most abundant cells, and these cells are alive at maturity. So they also have thin primary cell walls, and they also retain the ability to divide, which enables them to differentiate in response to injury or a changing environment. It is parenchyma cells, for example, that divide to produce the roots that emerge from a houseplant cutting placed in water. Parenchyma cells have vital functions, which include photosynthesis, respiration, gas exchange, and the storage of starch and other materials. So the cells that you see here on the slide is how parenchyma cells would look like under the microscope. Please take note of it, one of its most distinguishing feature, which is its thin primary cell walls. Next, we have collenchyma cells. Now, collenchyma cells are elongated living cells with unevenly thickened primary walls that can stretch as the cell grows. These cells provide elastic support without interfering with the growth of young stems or expanding leaves. The collections of collenchyma cells make up the tough, flexible strings in celery stalks. So, like what I mentioned earlier, take note also of the cell walls of collenchyma cells, because as compared to the parenchyma cells, which have thin cell walls, collenchyma cells are distinguished because of their, or can be distinguished by their uneven, thickened primary cell walls. Okay, next we have sclerenchyma. Now, sclerenchyma cells provide inelastic support to parts of a plant that are no longer growing. These cells, which are dead at maturity, have thick, rigid secondary cell walls that occupy most of the cell's volume. Now, the secondary walls typically contain lignin, which is a tough, complex molecule that adds great strength to the cell walls. Some sclerenchyma cells are elongated fibers. Linen, for example, comes from the soft fibers of the stems of the flax plant. Other sclerenchyma cells form hard layers or clusters, such as the small groups of cells that create a pear's gritty texture. So again, take note of how the cell walls of sclerenchyma looks like, because that is the easiest way actually on how you can differentiate parenchyma, collenchyma, and sclerenchyma from each other when you view them under the microscope. Now, this is how they are different when we compare them side by side. So again, parenchyma cells, they have primary thin walls or thin cell walls, and they also collenchyma cells, they have uneven thickened cell walls, while sclerenchyma cells have cell walls that consume most of the part of the cell. Next, we have conducting cells in xylem and phloem. Now, vascular tissues transport water, minerals, carbohydrates, and other dissolved compounds throughout the plants. And two types of vascular tissues are the xylem and the phloem. Now, xylem is also made up of types of cells, which are the tracheids and the vessel elements, while the phloem is made up of the sieve tube elements and the companion cells. In the next lesson, we will be discussing further the difference between a xylem and a phloem. Okay, so first we have the tracheids. Now again, the two kinds of water conducting cells in xylem are tracheids and vessel elements. Now first off, we have tracheids. So tracheids are long, narrow cells that overlap at their tapered ends. Water moves from tracheid to tracheid through pits, which are thin areas in the cell wall. So here you will see the pits that I'm referring to. So again, these are thin parts of the cell wall where water can move. Next, we have the vessel elements. Now, vessel elements are short, wide, barrel-shaped conducting cells that stack end to end, forming long, continuous tubes. Their side walls have pits, but their end walls are either perforated or totally absent. Now, water moves much faster in vessels than in tracheids, both because of their greater diameter and because water can pass easily through each vessel element's end wall. On the other hand, the narrower tracheids are less vulnerable to air bubble formation. So, if you look at the tracheids and vessel elements under the microscope, it would look like something like this. So, it again is easier to identify them this time based on their size. Next, we have the cells of the phloem. So first off, we have the sieve tube elements. Now, the main conducting cells of the phloem would be the sieve tube elements, which is aligned end to end to make a single functional unit, which we call as a sieve tube. The sieve tube elements are alive, but they lack a nucleus and they have little cytoplasm. So this is how the sieve tube element would look like. And when they are stacked up together, they form what we call as the sieve tube. Now, adjacent to each sieve tube element is what we call as the companion cell. So the companion cell is a specialized parenchyma cell that retains all of its organelles. Now, companion cells transfer carbohydrates into and out of the sieve tube elements, and they provide energy and protein to the conducting cells. So this is how the companion cells are attached to the sieve tube element. And if you look at them under the microscope, so this is how it is in a higher magnification. At a lower magnification, you will clearly see that the companion element is adjacent or found adjacent to the sieve tube element.